Hi, my name is Dr. Tycel Phillips. I'm an assistant professor at the University of Michigan, and I'm here to talk to you about a study I'm very excited about and enthusiastic and I think deserves your support, the repurposing of the BH3 mimetic venetoclax to treat newly diagnosed patients with mantle cell lymphoma. So to give you a bit of a background, mantle cell lymphoma is a rare non-Hodgkin's lymphoma, account for only 5 to 6%. The disease in itself is incurable, meaning the best we can hope for is to put the disease into remission at this current time. One of the biggest sort of needs in this area is to identify a treatment that is effective and can be safely given to all patients afflicted with the disease. Especially given that only 10% of patients will have what we call indolent disease, which tends to have the best outcomes and survival out of the other two subsets of this disease. Looking at the genetics, you'll see that cyclin D1 or the translocation 1114 is one of the first and key sort of pathognomonic signals in mantle cell lymphoma. But as you see following along the diagram, you'll see a multitude of other mutations that come along that will impact classic, blastoid, which is an aggressive variant, non-nodal leukemic, which is thought to be an indolent variant. And then you also have deletion of 17p, which can turn an indolent variant to aggressive one. Treatment currently today is based on chemotherapy, which is basically drugs that are designed to damage DNA, slash and burn, napalm. They're going to kill anything in its sight. So basically that accounts for a lot of the toxicities we see with chemotherapy, as it kills anything that grows very fast. So in addition, with mantle cell lymphoma, we don't have a standard of care. So treatment is dichotomized between patients who are either young fit, who get high intensity treatment, versus those who are unfit. Now, one of the keys to this high intensity treatment is we think and have shown in several studies that induces what we call molecular remission, which puts patients into a deeper remission, which has been shown to have longer progression-free survival and or remissions. So one of the keys that we need to do to improve outcomes in patients with mantle cell lymphoma is incorporate treatments which improve the body's ability to eliminate the cancer cells by targeting specific targets or proteins and reducing the side effects. So the biggest issue we have is the lack of equality. Again, as I said, older unfit patients don't get the best treatment because they just can't tolerate it. So what we want to do is design a treatment regimen using some of these new molecular targets we have for mantle cell lymphoma to design a treatment that's more applicable to all patients, will induce deep remissions, and again, lead to the longest remissions. So given the strides we've made in understanding the biology of the tumor, certain medications have come upon that are certain targeted treatments such as lenalidomide. So this study in conjunction with rituximab has been studied and approved in patients with relapsed refractory mantle cell lymphoma, but has been studied in newly diagnosed patients and been shown to be an effective treatment with response rates above 80%. The downside to this is that the treatment takes a very long time to reduce remission, so patients are still suffering from the disease. The treatment is given indefinitely, and also they didn't look at molecular remissions, relying solely on radiographic remissions. So what we want to do is to improve some of these aspects. We want to speed up the time to remission, induce deeper remissions, have the ability to stop actually the treatment and not treat patients indefinitely, and also lengthen the time patients can remain off of treatment. So to accomplish this, we want to add something to lenalidomide that doesn't sort of make the toxicity profile of the drug worse. So with this, we looked at the medication venetoclax, which is an oral BH3 mimetic. So basically how venetoclax functions, in the tumor cell, BCL2 sort of sequesters bad BAX, which are pro-apoptotic proteins, and prevents them from causing cell death. So venetoclax, when it's ingested, sort of mimics bad BAX and sort of binds to BCL2, which frees bad BAX to cause cell death. So with this, again, we have a different mechanism of action, a different toxicity profile, and something that can be synergistic with lenalidomide. Ongoing clinical trial using venetoclax with lenalidomide rituximab has currently enrolled 18 to 28 patients. To date, we've seen great responses. We've seen acceleration of time to remission with remissions within 3 months, molecular remissions within 3 months. We're seeing very little toxicity with no dose limit toxicity noted, and all patients basically tolerating this regimen to the date. And we have response rates of over 90+% in the patients we've treated. So what we want to do is further enhance and sort of needle down to how effective this regimen can be. So in this situation, what we want to do is expand this study to a phase 2. But in order to do that, we need to do further things. So one, we do want to add additional time points to look for molecular remission. We also want to further investigate the genomics of the tumor DNA to figure out, if we can determine who is responding, why they're responding, and the difference between the early responders versus patients who responded later. So currently the trial is being supported by the University of Michigan Clinical Trials Unit. We have a grant from the Rogel Cancer Center, and we also have funding from AbbVie. Additional funding from Cures will help us complete the enrollment of the clinical trial, add these additional time points to look for molecular remission, and perform this genomic test to look at home exome DNA of the tumor. Again, to help us identify the tumor cells, the factors of the tumors, which will help us to determine who's responding, why they're responding, and sort of speed of response. So due to these early favorable results, again, as I stated, we would look to expand this trial to add another additional 47 plus patients to formally compare to the Nordic regimen, which is our standard regimen of chemotherapy used in a multitude of institutions, which involves high-dose chemotherapy, inpatient and outpatient, autologous stem cell transplantation with 30 days in the hospital at least, and then maintenance rituximab. So the team itself consists of myself, Professor Phillips, I'm a lymphoma specialist, Dr. Melody Kendarpa, who's a lab technician who's also helping as far as a multitude of the preclinical work, which led to the development of this trial, and a multitude of tests as far as processing samples needed for the study. And Zanetta Nicolesa-Koleska, which is our BH3 expert, and who's performed the BH3 profiling that we looked to determine the sensitivity of the patient's tumor to venetoclax before starting treatment and while on treatment. So our expectation is this regimen will be designated as a valid off-label treatment by the National Comprehensive Cancer Network, which is a collection of comprehensive cancer networks in the United States, would put forth guidelines which are followed by insurance companies. We plan to increase this, complete enrollment of the Phase B study within 2020, if not for the current pandemic, which has affected our clinical trial enrollment. We feel we would have done completing this trial by now. After completion of the Phase 1B study, we plan to open and expand the Phase 2 expansion and would hope to complete that enrollment within the next 18 to 24 months, as we will add additional sites. Achieving the aims of this study with this additional expansion will allow us to substantially increase the number of patients able to truly receive effective frontline therapy, offer an improved safety profile compared to chemotherapy, and eliminate the needs of these patients to have undergo high-dose chemo and autologous stem cell transplantation, reduce the amount of time patients need to spend in a hospital, and overall decrease our financial burden on our health system. And with that, I'd like to thank you.